Good afternoon, and thank you for the introduction. My name is Yoshihisa Tanaka, and from Tokyo, Japan. Today, I'm going to present our recent work. This study aims to uncover how TDP43 dysfunction alters RNA splicing at a transcript isoform level by using short and long-read RNA sequence. As the previous speaker pointed out, the long and short read sequence have a good point and also bad point. So our work present you one good example to how these combining the short and long read provide a new biological insight. And I'm sorry, before I begin, I'd like to disclose that because I am currently employed by Daiichi Sankyo, a pharmaceutical company in Japan. However, this presentation is based entirely on academic research and is not related to the company's business or interest. Therefore, I have no conflict of interest to declare. Okay, here is the agenda for today's talk. I've structured my presentation into three main parts. First, I will introduce the background and the motivation behind our study. And second, I will walk you through the results, including our analysis pipeline and the identification of a novel cryptic exon. And finally, I wrap up with a brief summary and discuss future directions. Okay, let's start with the background section. This part is very, very important because without understanding this background, probably you don't interested in my talk. So I'm trying to understand you. And further, I also provide an overview of the design and goal of our study. Here, I will briefly introduce the concept of cryptic exons, what they are, and how they related to TDP43, and why they are important in the context of neurodegenerative disease. As many of you know, the RNA splicing is a fundamental process for generating diverse transcript isoforms. And these isoforms are critical for regulating gene expression and maintaining proper cellular function. Among the key regulators of splicing are RNA binding proteins. And today's central player, TDP43, is one of them. So TDP40 is an RNA binding protein. And usually, under normal condition, TDP43 localizes the nucleus, like this figure. And where it binds to an internemic region of pre-messenger RNA to ensure correct splicing. However, in 2015, a striking discovery was made. The knockdown TDP43 led to the aberrant inclusion of internemic region into mature messenger RNA. So these abnormally included internemic sequence are what we call cryptic exons. Then why cryptic? Because they are not supposed to be included in mature messenger RNA, and of course, they are absent from different transfer annotation like GenCode or RefSec. In other words, TDP43 plays a critical role in suppressing cryptic exons to preserve proper splicing. Now let's think about what happened when cryptic exons are included into messenger RNA. This inclusion is unexpected for cells and can have severe consequences, as illustrated in the lower part of this figure. This cryptic exon inclusion can introduce sometimes premature subcodon or lead to truncate or even toxic proteins. In shortly, the inclusion of cryptic exon into messenger RNA can disrupt the production of normal functional protein. So now let's finally move into a neurological context. In neurodegenerative disease such as ALS, it is well known that TDP43 becomes mislocalized to the cytoplasm from nucleus, where it forms a pathological aggregation. And this loss of nuclear TDP43 leads to the failure to repress cryptic exon and indeed the patient tissue shows the inclusion of cryptic exon in messenger RNA. Now, this is where TDP43 and cryptic exon and the neurodegeneration all come together now. This mechanism is now recognized as a key contributor to disease pathology, and it forms the foundation of our study. And especially the writing here, UNC13.2 and susmin2 have been demonstrated to have TDP43 dependent cryptic exons, and concept of restoring the susmin2 protein is now drug target for ALS. As we discussed, dysfunction TDP43 can lead to the inclusion of cryptic exon, which alters transfer structure, affects protein production, and may ultimately impair cellular function. To better understand these downstream effects, we reason that it's essential to capture the full-length transfer structures that contain cryptic exons. This background motivated us to perform long-read RNA sequencing, which theoretically enables us to observe how cryptic exon inclusion reshapes entire transfer isoforms. In our study, we aim to leverage this approach to uncover previously unrecognized transfer alterations caused by TDP43 dysfunction. As materials to mimic the TDP43 dysfunction in neural context, we differentiate iPS cells into motor neurons and knock down TDP43 using shRNA. Now, I walk you through the key result of our study. Before performing long-read RNA sequence, we began by analyzing short-read RNA sequence data from iPS-derived motor neurons with TDP43 knockdown. Short-read RNA sequencing is a particularly good tool for detecting a local splicing change very accurately, owing to its high read depth and base level resolution. And after sequencing, we perform a differential splicing analysis between the scrum control and TDP43 knockdown samples. And from the analysis, we finally identify 53 cryptic exon candidates in total. So we then use these 53 candidates as a focus of our long-read RNA sequence experiment. And to capture the full-length structure of transcript containing cryptic exons, we next perform long-read RNA-seq using the nanoparticle platform. The sample are the same as in the short-read experiment, and that is iPS-derived motor neurons. And the problem is happen here. However, we quickly encounter a challenge here. Which tool should we use to analyze the long-read data? In recent years, many tools have been developed for analyzing long-read RNA-seq data. For example, here, SlingTie2, Bamboo, RNA-Bloom2, FLAIR, IceQuant, and Espresso, and more. But currently, there is no gold standard tool, especially for reconstructing a complex or aberrant transfer isoform. This is a hard task. I'm showing one study here supporting this. This benchmark study was published last year in Nature Method. And the main point here is that they say that the sensitivity of most tools is quite low, described as this upper figures. And that is particularly for identifying the transcripts that are not present in the difference annotations. Since cryptic exons by definition are not annotated, right? This low sensitivity is absolutely a problem for our study. So we think that existing long-read RNA tools are not sufficient for our study. So that is why we decided to develop a new tool. Let me briefly introduce IsoRefiner, the tool we developed for this study. IsoRefiner is not based on the novel algorithms, but rather use an ensemble approach. Main idea is to combine the strengths of existing tools. IsoRefiner independently runs several widely used tools such as here, RNA-Bloom2, Bamboo Espresso, IceQuant, and SlingTie2, and then merge their output to generate a more comprehensive transfer isoforms. The workflow is very, very simple. The input is a aligned long-read bound file, and output is a GTF file, including the reconstruct transfer isoforms. The one unique aspect is that we include RNA-Bloom2, which is a de novo transfer assembler, unlike other tools. Data is not shown here, but in our analysis using simulated dataset, this simple but effective strategy improves sensitivity compared to using individual tool alone. For this study, we select 5 tools written here, but user can have flexibility to select which tool to run based on your computational resource, wherever you like. And we apply isoRefine to our long-read data, and we successfully reconstruct transfer structure containing cryptic exon in 7 genes written here. And that means is a positive control. And our analysis show that isoRefiner and iRNA detect the highest number of cryptic exon containing transcript from long-read data. And you might see that iRNA appears similar to isoRefiner, right? But problem is here, the iRNA produce an extremely large number of transfer structure, so which made me it's very hard to find the transfer isomer that we want. This suggests that iRNA have a higher rate of false positive compared to the isoRefiner. This is our advantage. So among the 7 genes we identified, we focus on MNAT1 because it was previously unreported and show a highest level of cryptic exon inclusion. On your left side of slide, you can see the full-length structure of MNAT1 containing the cryptic exon. Importantly, this structure was obtained only from long-read data. The cryptic exon is located between exon 5 and 6. On your right side, the top one shows the cryptic exon signal, this one, appearing only in the knockdown sample, and this is not present in the gene code here. And the bottom panel shows the transfer structure reconstructed by various tools. And notably, both bamboo and string titles failed to reconstruct the correct isoform containing cryptic exon. Probably this is a very good lesson from that we need to learn. I think we need to, this highlight the risk. This lessons tell us the risk of relying on the single analysis tool, which could lead to incomplete or incorrect conclusion. We next analyze the profile of the MNAT1 cryptic exon. On your left side figures, this cryptic exon is 32 base pair long and flanked by a canonical splice site following the splicing rule AGGT. Importantly, we also identified the TD46 binding motif, TG repeat here, just upstream of the cryptic exon. To validate the cryptic exon experimentally, we perform a reverse function PCR using 2 custom primer sets. As expected, the cryptic exon specific primer produced a band only in the knockdown sample, and the canonical exon spanning primer showed a larger band corresponding to the inclusion of cryptic exon. These results provide experimental support for the presence of the MNAT1 cryptic exon. And you might know that the cryptic exon is 32 base pair long. And probably some of you guys might notice it. 32 is not divided by 3, right? This suggests that frame shift occurs upon its inclusion. Indeed, we found the inclusion of the cryptic exon into this premature termination codon here in exon 6. Since the PTC is followed by downstream exon-exon junction, this transcript is likely targeted by the nonsense mediated decay pathway. To test this, we examined the expression level of MNAT1 using short read data. As we expect, MNAT1 expression was significantly reduced in the TD43 knockdown condition. These findings suggest that inclusion of the MNAT1 cryptic exon induced a premature termination codon, which in turn leads to transfer degradation and reduced gene expression. And finally, we assess clinical relevance of MNAT1 cryptic exon. We analyze brain tissue sample from ALS and FTD patient to determine whether the MNAT1 cryptic exon is preserved in vivo. Surprisingly, we detect the cryptic exon at the exactly same position in patient sample. And this is supported by quantitative analysis of cryptic exon inclusion in your right side. This indicates that the splicing event exists not only in vitro, but also in human disease context. While further validation is required, these findings indicated potential involvement of MNAT1 in TDP-43 proteinopathy. Okay, I will summarize our finding and discuss potential future directions here. To summarize, first, we show that combining approach using short and long read RNA provide a powerful strategy for analyzing translation change, especially for capturing aberrant translation structure like cryptic exon. And we developed IsoRefiner, new analysis tool that outperforms existing long-read tool in detecting especially novel isoforms. And third, using IsoRefiner, we discovered a previous unreported cryptic exon in MNAT1 in TDP-43 nocturnal motor neurons. And fourth, importantly, we also detect this cryptic exon in brain tissue from the patient, underscoring its potential role in TDP-43 proteinopathy. And lastly, I think that this strategy could be broadly useful for studying disease where splicing plays a key role and may support the development of new diagnostics or therapies. And this is my final slide. And this, our developed IsoRefiner is available, freely available at GitHub, and you just type conda install IsoRefiner. And also this study has been published at Communication Biology last week. So you can, if you want more information, you can read to my paper. Thank you very much and your time and attention. Thank you.
Thank you. Yes.
Hi, thank you for a nice talk. So I have questions about IsoRefiner because you are combining different tools.
Okay.
But at the end of the day, many of these tools are using the same mapper. And I was wondering whether, some biases introduced by the mapper could be also responsible for this lack of detection in some cases, and whether you have considered also including the assessment of additional mappers into your pipeline.
Yeah, I think that would be a good idea to include that, but I guess very important point is include the de novo assembly tool here. De novo assembly tool. That is a very important, I think.
Okay, thank you.
Any other question? Maybe last one? Okay, if not, we're going to thank you so much.
Thank you.